I don't think there's much of a difference from a manufacturing perspective, whether we want to address solid or liquid tumors. I think cell therapy is still a nascent space. We're still in the process of identifying the right tools that can consistently produce cell therapy drugs. Where we do have a difference, though, and I think that's important to know, is solid tumors are very different in the way that they are configured. They're very, very tightly packed cells. They often have a very different extracellular matrix composition, and so really getting into these tumors is difficult. So the field has been trying out CAR-Ts because they have been really successful in liquid tumors, of course, but haven't really been able to see the same clinical results. So that's where the difference comes in now, because we're trying to find different approaches to using cell therapies, but evaluating different types of gene editing of these of regular CARs, or regular T cells, I should say. But at the same time, we're also trying to evaluate if other cell types in the immune system might be better at penetrating these solid masses. So we're looking into TILs, tumor infiltrating lymphocytes, I think, NK cells, natural killer cells, macrophages. So there's a host of different cell types that are currently being evaluated. And I still maintain that I think the platform technologies that we're developing right now, like our ECHO, other technologies that are coming into the space, can serve all of these cell types, but we will have to tweak. So cell selection will be different. It's not a CD4, CD8 necessarily. It might be a CD56 or something, right? So we will have to adapt and optimize our cell selection criteria. We will probably use different CARs. So they need to be developed and evaluated, because these solid tumors may have a different signature. And then we're also obviously, solid tumors are a much larger group, so the patient pool is much, much bigger. So we do need to upscale somehow, right? And the easiest way is to go allogeneic off the shelf. For that, we need to gene edit, we need to manipulate the cells differently, and I think that's still where we're trying to figure out how best to do that, how most efficiently to do that. And then obviously to upscale and make larger quantities while maintaining cell health. So there's a lot that we can learn and that we can borrow from CAR-T, and then I think there's tweaks and adaptations that will come into play.